Hi everyone, thank you for joining us today. My name is Chris Reed and I'll be talking to you about digital PCR and the RainDrop Digital PCR System. First off, what is digital PCR? Digital PCR is simply a way to take a sample, divide it over a very large number of reaction vessels, PCR those reaction vessels, and then count how many of those reaction vessels show amplification of your target. This is a way of doing what we call needle in a haystack detection. If you have a very rare target that's present in a high background, if you spread that sample across a very large number of reaction volumes, your target is going to be spread across those reaction volumes as well as your background, and your target is going to be present at a higher concentration in those reaction vessels compared to the original sample. Now that panel in the middle that says divide is showing only the target molecules. We're not showing the background molecules. But please remember, all of those droplets that say non-target droplets do have background molecules in them. The important thing to remember is your target molecules will end up individually in those droplets, and because they are at a higher concentration, it makes it easier to detect them. So what sort of experiments would you have where you would need to do needle in a haystack detection? When would you need to use digital PCR? Well, if you have too few mutations and too high a wild type background, you need to do digital. If you have too little fetal DNA and too much maternal background, you can do digital PCR. If you have too few viral particles and a background of a lot of tissue or blood, digital PCR can help. It can also help if you have very few copies of your gene target and you're running for too many PCR cycles. If you look at the amplification plot at the bottom of your screen, you can see at the early PCR cycles where you have high concentrations of your target, your replicates give very good agreement with each other. However, when you get up to the high CT values where you have low copies of your target, you see a broader spread between your replicates. This is the area where digital PCR can help you accurately quantitate your targets. We're used to thinking of digital PCR, or we're used to thinking of PCR as taking place in tubes or in 96 or 384 well plates. Using RainDance technology, we can produce millions of droplets. Each one of those droplets is equivalent to a well or a tube. So your PCR is taking place inside millions of droplets instead of single well or 384 well plates. What sort of applications is it useful to be able to do digital PCR and count your number of target molecules? Examples are rare mutation detection, copy number variation, viral load, fetal DNA detection, and many others. This is an example of how you can count single molecules in digital PCR. So your sample is going to start out with background molecules, which we don't show here, and also your target molecules. You're going to mix those with your assay and your master mix. Then you're going to spread them across that very large number of droplets, millions of droplets. Now again, we're showing droplets here that don't have any target in them. They still do have background material. The droplets that have the target molecule in them have the target molecule present at a higher concentration, which makes them able to amplify and be visible above the background. Also, because we have so many partitions, you're going to end up with target molecules alone in the droplets, which allows you to do single molecule counting. After you partition your sample into the droplets, you then PCR amplify those droplets. After the PCR amplification is done, you're going to read the total amount of signal after PCR is complete, and you'll be able to detect which droplets showed amplification of your target and which ones did not. At the bottom right-hand corner of the screen, you can see a scatter plot, which is showing two different clusters. One of them is the PCR negative cluster, which is just showing you the droplets that did not have PCR amplification, and a second cluster that is showing amplification in the VIC channel, and since we use a VIC probe, those are the droplets that are positive for the target we are looking for. So this is doing just a single-plex reaction. However, many people want to be able to do a duplex, where they want to look for something, for example, as a mutant allele present in a high concentration of wild-type background. In that case, it would be useful to be able to detect both of those assays, both of those targets, in the same sample at the same time. And that would require multiplexing. Multiplexing is often considered to be very difficult in real-time PCR. If you think about what you're doing when you multiplex, you have a tube that has all of the different targets that are present. You have two or more assays, and you want them to amplify their respective targets individually without competing with each other, even though they're trying to use the same pool of DNTP and enzymes. This can cause competition of reagents. You also can have inhibition of the primers and probes competing with each other. For this reason, multiplexing in qPCR can be difficult and is often avoided. Multiplexing in digital PCR is done in a very different manner. In this case, each of your droplets are going to end up containing all of the primers and all of the probes for each of your assays. But all of the droplets do not contain all of your target molecules. Because we're spreading our sample over a very large number of millions of droplets, your target molecules are going to end up by themselves in the droplets. So you can see we have one droplet that has just a target molecule for the Vic assay. In that case, the Vic assay would amplify, but there are no targets for the SAM assay, so there's no competition taking place in that droplet. A separate droplet would have a target molecule for the SAM assay. Likewise, there wouldn't be a target present for the Vic assay, therefore you don't get that competition. This is an example of what data would look like when you're running a multiplex. Again, we have a Vic-labeled probe detecting our wild-type target and a SAM-labeled probe detecting our mutant. If you look at the scatter plot on the right-hand side, you can see we have, again, a non-amplified cluster. We also have a cluster that's amplifying in the Vic channel, where you can see the number of droplets positive for your wild type, and also a cluster in the SAM channel showing you the number of droplets that were positive for your mutant assay. So why is it important to have 10 million droplets? Again, if you think about the sample that you're running, you have target molecules generally present, very, very low copy number, but there's lots and lots of background molecules that are present. Again, these droplets that we're showing look like they're empty, but they do have background molecules present in them. Your targets are going to be spread out over this very large number of droplets, such that you have fewer than one target molecule per droplet. This means the target molecules are most likely going to be alone in that droplet. When you do your amplification, you can count the number of positive droplets, and you'll know how many target molecules you had. In this case, we're looking at just 20 droplets. We had 2 that were PCR positive, and that means we had 2 target molecules. What if you don't have enough partitions? What if you can't make 10 million partitions? In that case, when you spread your sample out across your partitions, you may end up with more than one target molecule per partition. In that case, if you just count the number of positive droplets, you're going to underrepresent the number of target molecules you have. Again, in this example, we're looking at 20 droplets. We have 1 negative, we have 19 positive, but we have 32 different target molecules in there. If you don't have enough partitions, you need to use Poisson distribution statistics to estimate the total number of target molecules that you have. Something else that you can get from 10 million droplets is the ability to simultaneously detect very large copy numbers and very large copy numbers of the targets that you're looking for. In this data example, if you look at the top panel, we are looking at 2 million positive droplets for EGFR wild type and no positives for the mutant. In the bottom panel, we are again looking at 2 million positives for the wild type. And if you look at the blow-up of the mutant area, you can see there's 303 mutant droplets there. This means we're detecting millions of droplets and hundreds of droplets for the same sample at the same time. With our platform, we've also demonstrated a sensitivity as low as 1 mutant in 250,000 wild-type background molecules. 10 million droplets also gives you a large dynamic range. The RainDrop Digital PCR System is linear over 6 logs. This, as I said before, enables the measurement of low and high copy number targets at the same time. It also eliminates the need to dilute your sample to find the sweet spot, which is the best concentration to run your sample at. And because we're getting true single molecule measurements, it allows us to do multiplexing. This particular customer happened to be looking at mutations in the KRAS gene in colon cancer, but KRAS mutations are also found in other cancers such as leukemia, pancreatic, and lung. KRAS mutations can be predictive of resistance for certain chemotherapy agents. And 90% of the activating KRAS mutations occur at 7 different positions and 2 codons in the gene. Now, as I said before, you really want to know how many mutant targets are present relative to the number of wild-type molecules. But to look at 7 different mutations would require running that sample 7 times, 1 for each pair of assays, mutant 1 in wild-type, mutant 2 in wild-type, etc. So it would be really useful if we could develop a way to look at more than just 2 different assays at the same time. That was where we got the idea of multiplexing, not just with color, but also with the intensity of the probe. So we discussed color already. You can use 2 different dyes, such as SAM and Vic. However, you can also multiplex with intensity. Using multiplexing with intensity, you're using 2 different assays labeled with the same dye, but you're using a higher concentration of one of the assays, which allows it to have a cluster further away from the non-amplified cluster, and you can optimize so that you have good separation and good clustering of those particular assays with each other. And this would allow higher order multiplexing. So this is an example of data where we did multiplexing with color. You can see target 3 is VIC labeled. And then if you look at the FAM channel, we actually have 2 different targets, both labeled with FAM. These are different assays that are detecting different targets. One of them has a high concentration of FAM. The other assay, we use half the amount of the FAM probe. And as you can see, we get 2 very distinct clusters that are well separated, and they're very easy to tell apart. But we have a lot of real estate left on that 2D plot, and it would be useful to be able to fill that in. That was where we got the idea of taking assays, making a probe, and labeling part of that probe with FAM and part of that probe with VIC. By doing that and then varying the concentration of the assays, we were actually able to make this into a 5-plex. You can see we had 1 probe that's labeled with VIC, 2 labeled with FAM, and then we had 2 other probes that were a mix of FAM and VIC with different concentrations of the FAM. And this allowed us, again, to make this assay into a 5-plex. So getting back to the KRAS mutations, we were able to take those 7 mutants plus the wild-type assay and split them into 2 panels. The panel on the left-hand side has the wild-type assay plus 3 of the mutant alleles that we were looking for. The panel on the right has the KRAS wild-type and 4 different mutations. So instead of running each sample 7 times, we only had to run each sample 2 times. I do want to point out that these are control DNAs. You hopefully would never see a patient with that level of mutant alleles present. By running positive controls, we were able to determine where the clusters would be for the particular mutations, so that when we did run patient samples, we were able to determine which mutation they were positive for. Now I'm going to talk a little bit about the RainDrop platform in particular. So the RainDrop platform has a very simple workflow that requires just 1 pipetting step per sample as you go through the process. You begin by loading your sample onto the source chip. The source chip is then placed into the source instrument, and that's where the droplets are generated. You also place an 8-strip PCR tube into the source instrument. As the emulsion is generated, it flows into the PCR tube. When the instrument is done, you cap the PCR strip, you put it in a PCR machine. When the PCR is complete, you put the strip tube into the sense instrument, place a chip on it, and it will read how many fluorescent droplets you are. Please note there's 1 pipetting step throughout this entire process, and once the droplets are generated and the caps are stripped, there is no reason to remove those caps, and you will have very good contamination control throughout the process. We also have analysis software called RainDrop Analyst that has a very simple workflow. You simply take your sample files, drag them into the RainDrop Analyst workspace. It will generate a 2D scatter plot for you. You define your cluster, and the software will tell you how many droplets are contained in the cluster that you're looking at. The data can then be exported into Excel. Many people ask, how easy is it to transfer my existing qPCR assays into digital PCR? People often have real-time assays that they've been using for a very long time and would like to continue to use them, but get the sensitivity that you can get from digital. And these are just some examples of assays that people have asked us to work with. Out of 53 total assays that we have, real-time assays that we've been given that we've tried in our digital PCR system, 49 of them worked immediately with no optimizing required. The other 4 we were able to optimize by simply modifying the annealing temperature. Another question we get is, what master mix do I need to use? We don't have a proprietary master mix, and customers use generally the same master mixes they use in real-time PCR. This is a list of master mixes that we've tested in-house. You can see from a number of different companies, a number of different types of master mixes, and we do have recommendations available on request. Another question is, what sort of probe chemistries do we recommend? And we have, again, tested a number of different probes from a number of different companies, including Pac-Man MGB probes and camera quench probes from Life Technologies, assays from BioSearch and Sigma, and also primetime probes available from IDT. And in fact, we have a partnership with IDT. If you look at their July 2013 issue of Decoded, you'll see an article featuring the partnership between RainDance and IDT. So here's an example of how that partnership has benefited us. One of the assays that we were asked to look at was the T790M mutation for EGFR. Now, this is a very difficult assay to develop. There's a 10-base repetitive element that's repeated twice in the probing region. The red SNP is actually the target, and there's another SNP upstream of it that you want to avoid when you're doing genotyping. And in addition, this entire region has 83% homology to another region in the genome. So trying to develop an assay that is specific for this particular SNP without picking up anything else can be very difficult. There are qPCR assays available that can detect one mutant in a wild-type background of about 100. Taking one of those real-time assays, we placed it onto the RainDrop system and ran it with no optimizing of any sort. And we were able to detect one mutant in a wild-type background of 5,000. So it was a drastically improved precision just by taking that assay from real-time and running it in digital. But as you can see from this data, it definitely could be improved. The clusters don't have good definition, they don't have good separation, and the amplitude of the signal is not very high. So we worked with IDT to design a custom LNA-Zen quenched probe, and as you can see from these plots, the data drastically improved. And we're able now to deliver a sensitivity of one mutant in a wild-type background of 11,000 molecules. And if you look at the panel on the left-hand side, on the right-hand side, there are 3.5 million negative droplets. We were also able to see 43,000 wild-type droplets, and we were able to see as few as 8 mutant positive droplets. I'm going to describe now some of the projects that we've worked on with our customers. So many customers are working with genomic DNA, and we have worked with genomic DNA from a number of different sources, including DNA from cell lines, from fresh frozen tumors, from FFPE tumors, circulating tumor cells, and we've also done some work with methylation quantitation in genomic DNA. And this is an example of working on copy number variation. So we were looking at a disease called Spinal Muscular Atrophy, and this was published in Lab on a Chip in 2011. The gene that controls spinal muscular atrophy is called SMN1, and if you don't have any copies of SMN1, you will develop spinal muscular atrophy. However, there are 4 different phenotypes for this disease, and what phenotype you have is modified by how many copies of the SMN2 gene that you have. So if you look on the right-hand side of the left-hand side of the screen, you will see this is the 5-plex I talked about earlier. In the SAM channel, we have an assay that will count how many copies of SMN1 and SMN2 are present. And the top, we have those other 3 assays. 2 of them are for mutations, point mutations that are in the promoter region, and the one in the middle, the BTK-DHA, was a reference gene that we used to control for the amount of genomic DNA that was present. If you look at the left-hand side of the screen, you can see normal controls. And if you just look at the green bars and the normal controls, you can see that everybody has 2 to 3 copies of the SMN1 gene. This is why they're normal. They have the proper number of copies. If you look at the SMN2 gene, which is the red bars, you can see there's quite a bit of variation in the copy number present, even in the normal controls. Some people have 1. We've got a person that has 0, and some people have 2. We are also able to identify a carrier. If you look at the carrier individual there and look at the green bar for SMN1, you can see that they only have 1 copy. The reason they're a carrier and don't actually have spinal muscular atrophy is because if you look at the number of SMN2 genes that they have, it's got 5. On the far right-hand side, you can see there are 4 different affected individuals. All of them have 0 copies of SMN1, and they have varying numbers of SMN2 genes, which is what is modifying their phenotype. We've also worked with a number of customers that are looking at circulating cell-free DNA, but we also have people that are looking at cell-free DNA in several different cancers, including breast, colorectal, and pancreatic. And this is an example of looking at KRAS mutations present in metastatic colorectal cancers. This is using that 7 mutation panel and 2 different panels that I talked about earlier. So in the original study, they had 95 patients that had metastatic colorectal cancer, and they used regular real-time PCR to screen the tumors for KRAS mutations. Of those 95 patients, 27 of them had tumors that were positive for KRAS using traditional real-time PCR. We then took those tumor samples and reanalyzed them using our RainDrop Digital PCR panel. When we did that, the 27 patients that were positive for KRAS with real-time PCR were verified with digital. So the 27 that were positive with real-time were also positive with digital. But interestingly, we also identified 2 other mutations that some patients had that were missed by real-time PCR. We also looked at the 68 patients who had KRAS negative tumors by real-time PCR, and we actually were able to detect 18 of those patients had KRAS mutations present. If you look at the percentage of the KRAS mutations, you can see they fall into several different populations. On the left-hand side, we're looking at just the tumors that were positive by real-time PCR. And you can see that they had, on average, about 35% of their tumor contained KRAS mutations. If you look at the ones that were negative with real-time but were positive with digital, so these are the 18 that were positive only with digital, you can see that it was, on average, 9% with quite a bit of variation there. Of the patients, of the tumors had, the cells in the tumor had KRAS mutations. But the interesting thing here is this is actually kind of divided into 2 different populations. One population had 1% of more of their tumor that was positive for KRAS, but there were a small number of patients that had a very small number of cells that had KRAS mutation, less than 0.1%. This is a new clinical population that hasn't ever been identified before, so it's very interesting to see what their long-term prognosis will be. After looking at the KRAS mutations that were present in the tumor, our customer wanted to know, would we be able to pick those mutations up by just simply taking a blood sample and looking at the cell-free DNA present in the plasma? So in this study, we had 54 patients that had metastatic cancer, and 19 of them had tumors that were positive for KRAS using real-time PCR. We then collected blood from those patients, and we used digital PCR to test the blood samples in the plasma of those patients for KRAS mutations. For the 19 patients that had tumors that were positive for KRAS, we were able to identify the same KRAS mutation in the bloodstream of those patients. In addition, there was another patient where we identified a different mutation. It wasn't the mutation that was seen in KRAS, but we did pick up a KRAS mutation. And for 2 of the patients that had KRAS positive tumors, when we tested their plasma, we were unable to pick up any KRAS mutations circulating in their bloodstream. Also very interesting is for the 35 patients that were negative, their tumors were negative for KRAS using real-time PCR, we were able to pick up 2 patients that actually did have circulating KRAS mutations. The last set of samples that we've worked with are cDNA. Working with cDNA, we have done mRNA expression profiles. We've looked at the expression of the gene fusion transcripts. We've looked at exosomal mRNA, and we've also looked at the expression of miRNA. And this is an example of miRNA biomarker assay with multiplexing. So there are just a couple of data points here, but I find it interesting because we were able to develop a multiplexing assay with an off-the-shelf miRNA assay that was labeled with FAM. So if you look here, we have 2 different. One patient was normal, and when he was assayed with the miRNA assay, there were actually no positive droplets that we saw. That was affected when they were assayed with the 2 miRNAs, you can see we had 2 different clusters that had definite high amounts of positive droplets in them. And as I said before, these were off-the-shelf microRNA assays, both labeled with FAM. We were able to multiplex them by simply adding twice as much of one of the assays, which allowed us to have 2 different clusters and allowed us to double the throughput when looking at our assays. So what could you do with 10 million droplets? You could possibly detect even rarer alleles in tumors and do liquid biopsies. You could look at even smaller changes in an IPT, which could mean looking at and detecting duplication or deletions of autosomes in fetal circulating cells in maternal blood. You could look at very low levels of copy number variation and potentially pick up somatic mosaicism. You could also look at very rare pathogen or viral loads, which allow you to detect patients earlier in infection and also detect low level of residual disease. And I'm sure everybody has an additional idea they could think to add to this list. So in summary, advantages of the RainDrop Digital PCR System. Because we have up to 10 million droplets, we can have true single molecule counting. This gives us very high sensitivity for very rare events. Single molecule detection also allows us to do assay multiplexing. Our closed tube design allows us to minimize contamination, and our one pipette process is a very simple workflow. And the applications that we've demonstrated are rare mutation detection, copy number variation, viral load measurement, and quantitative expression, miRNA, and others. Thank you for your time. Are there any questions? So we have a question here that says, is the RainDance system available in Germany and Europe? If yes, is there a sales rep whom I can contact for further information? We actually have, we can capture your email address here, and we can have your sales rep contact you. Another question that we got is, can you use only SAM or VIC labeled probes? You can use probes that have dyes that emit in those particular wavelengths. It doesn't need to be exactly SAM or VIC. We have customers that instead of VIC are using PET or MAX or sometimes Yakima yellow. So there are other options for probes that you would like to use. We had another question, what is the lowest concentration of DNA that the RainDance can detect? We have run very small down to, fraction of picogram DNA. Really, kind of when you get down to that level, it depends on, do you end up with any target molecules in there and the sensitivity of the assay that you're running. We had another question, is what is the sample volume per droplet? The droplets are generated with a 5 picoliter volume. The total reaction volume that you can start with can be as, when you generate your droplets, can be as low as 25 microliters or as high as 50 microliters, but the droplet size is 5 picoliters. We had another question, how many dyes can the instrument detect? Right now it is a 2-channel, 2-dye system, but by using the combination of fluorescent intensity and different dyes, we can multiplex. We have demonstrated up to a 10-plex with a single sample. We had another question, how long does it take to generate droplets for 8 samples? The droplets are generated in parallel with each other, so it takes 30 to 35 minutes to generate the droplets for the 8 samples. They're all done at the same time. We had another question, do you need to use probes or could you use intercalating dyes? Right now we only support probe technology on the RainDrop system. We had another question, is there any cross-contamination between droplets or merging at high temperatures due to surfactant degradation? We don't have any cross-contamination of droplets. The droplets are all generated in their own individual channels and then are placed in individual PCR tubes. We also had a question, are there any publications on dPCR that I can refer to? If you go to our website, which is www.raindancetech.com, and go to the dPCR tab, there is a tab that lists publications that are available using our system. Okay, that looks like all the questions we have. Thank you for your time, and everybody have a good day.